Have you ever looked up at the night sky and wondered how the vast expanse of the universe, with its countless stars and galaxies, came into existence? How do you explain the incredible diversity of plants and animals that fill our habitable planet? How old is the Earth? And how can we know for sure? And how do you explain the origin of mankind and the beautifully diverse ethnic groups? Put simply, where did everything come from?
For centuries, we have struggled to understand where we came from. Many theories have been proposed, some rational and thought-provoking, others wildly speculative and seemingly impossible to verify. So how do we sort through all of these theories to determine which one is most logical? Modern science is revealing important clues to this puzzle, helping us to better understand the full picture of our origin. However, the mystery still remains. Where did everything come from?
The standard answer offered is the Big Bang explanation for the origin of the universe and the evolutionary theory for the origin of the species. This means whether it was the formation of the universe, the first life forms, or people, the answer is the same. It was all the result of blind natural processes acting on matter and energy over many millions and even billions of years. As stated by National Geographic magazine, it all began in chaos. In other words, the universe and everything in it, including you and me, are nothing more than the sum total of a series of natural accidents. For the Big Bang theory, the universe formed through accidental collisions of atomic particles. For biological evolution, the species formed through accidental copying errors in the DNA called mutations. This is the story we've all been told. But what if the standard answer is not as convincing as we may have been led to believe? What if there are fundamental problems with the Big Bang and the evolutionary theory that scientists are well aware of, but have not been made known to the general public? In the last decade alone, compelling new evidences from the major fields of science have cast serious doubt on the evolutionary view of natural history. Discoveries so profound, if made known to the world, would have the power to drastically change our understanding of the past. Might the biblical view of origins as described in the book of Genesis offer a more reasonable explanation? To some, it is extremely naive to even consider the Genesis account of creation on the grounds that it is believed to be nothing more than a primitive myth, and therefore totally incompatible with modern science. This stems from the widely held perception that creation is religion and evolution is science. But is this really true? Is it proper to equate evolution with science? Does science have the ability to address questions regarding past events that we were not there to directly observe or verify? Events like the spontaneous origin of the universe, the origin of life from non-life, and the evolution of the earliest life forms into mammals. Or might we be giving science a power it does not have? To answer this, it is important that we accurately define science as well as its limitations. When we think of scientists in white lab coats analyzing cells under a microscope, or chemists mixing acids and bases, it is known as observational science. This is the type of science that led to finding cures for diseases and our advanced technology: computers, smartphones, orbiting satellites, and Mars rover robots. A good definition of observational science can be found in standard introductory level biology textbooks, which state, "Scientific inquiry is a powerful way to know nature, but there are limitations to the kinds of questions it can answer. These limits are set by science's requirements that hypotheses be testable and falsifiable, and that observations and experimental results be repeatable." Notice observational science has definable limitations to the kinds of questions it can answer. Hypotheses must be based on observations and experimentation. In addition, hypotheses must be testable and their results repeatable. Observable, testable, and repeatable experimentation are the hallmarks of good observational science. When biologists grow bacteria on a nutrient medium to study adaptations, growth rate changes, or sequence their DNA to determine which genes suffered mutations, they are practicing good observational science. But answering the question of where bacteria came from to begin with, their origin, is outside the bounds of observational science.
Science really is about the study of the operation of the present world. And a lot of people think that, well, no, some science you study the past. Well, not really. I mean, when you dig up a fossil, people say, see, there you go, you're looking at the past. Well, no, you're looking at the present. Fossils exist in the present. And that's not to say we can't make some guesses about the past based on some of the scientific evidence we see in the present, but our worldview, our way of thinking, our view of history will obviously influence any reconstructed past. Same goes with stars and galaxies. People say, you look at a galaxy, you're looking back in time. Well, I'm looking at the galaxy today, right? The photons are hitting my eyes today, and so astronomy really takes place in the present. And granted, we can speculate about how galaxies looked, 1,000 years ago, or what stars looked like thousands of years ago. But the fact is, those kind of claims are not scientifically testable. The kind of science that makes computers work and puts people on the moon, real science, observational science, is testable and repeatable in the present. It doesn't take place in the past, it takes place now.
According to Big Bang cosmologists, the universe began 13.7 billion years ago. The Earth is said to have formed 4.5 billion years ago. The first fish evolved 530 million years ago. Dinosaurs evolved 230 million years ago. Mammals 120 million years ago. The earliest primates 50 million years ago. And Homo sapiens evolved from hominins roughly 200,000 years ago. Realize the most significant evolutionary events all allegedly occurred in the unobservable, unrepeatable, untestable, and therefore unverifiable past. Almost the entire evolutionary theory, including the Big Bang, is outside the realm of observational science. All of the aforementioned past events, and many more not mentioned, are unamenable to the scientific process. No man was there as an eyewitness to provide for us first-hand knowledge of what actually took place. Thus, evolution is more appropriately a history. Ernst Mayr is hailed by the scientific community as the Darwin of the 20th century. As a distinguished evolutionary biologist, Mayr was more than qualified to make the following admission: that evolution is a history, not an observational science. He affirmed this, saying, "Evolutionary biology, in contrast with physics and chemistry, is an historical science. The evolutionist attempts to explain events and processes that have already taken place. Laws and experiments are inappropriate techniques for the explication of such events and processes. Instead, one constructs a historical narrative, consisting of a tentative reconstruction of the particular scenario that led to the events one is trying to explain." Here Mayer did not place evolutionary biology on equal planes with physics and chemistry, which are clear examples of observational science. Instead, he describes evolution as a historical science, a very inexact type of inquiry, the tentative reconstruction, meaning it is subject to change whenever the theorizer feels it is necessary to better fit the evidence. When evolutionary scientists attempt to explain what they think happened in the past, they first presuppose that the evolutionary view of history is correct. This view of history then serves as a framework from which all evidence and data are interpreted. Mayer described this presupposed view of history as a narrative, literally a story written by scientists based on what they believe happened in the unverifiable past. The biblical view of origins, as described in the early chapters of Genesis, chapters 1 to 11, is also considered a historical narrative. The Genesis account of creation is not written in a style typical of Hebrew mythology or poetry. Instead, it is recorded as a sober historical account with precise chronological reference points, carefully detailing major origins-related events, including the recent creation of the heavens and earth, the first male and female, a global flood, and the formation of the various people groups and languages. The implications of this are profound. Rather than an issue of science versus faith, the creation-evolution controversy ultimately boils down to a face-off between two opposing histories. The major difference is that the evolutionary view of history was narrated by scientists within the last 150 years and is tentative to reconstruction, whereas the biblical view of history was recorded thousands of years ago and is not subject to continual revision.
Some might object to accepting the origins issue as a battle between two different histories and insist it actually comes down to a matter of evidence. You may think, do we really need to presuppose a certain view of history in order to accurately address the question of origins? Wouldn't it be better to just let the evidence speak for itself? While this may sound like the most objective way to approach the origins issue, it is based upon a fundamental misconception about the nature of evidence, called the evidence scale fallacy.
A lot of people have the impression that the way that you resolve the origins debate is that you try to accumulate the most evidence, as if there's a scale and, you know, the evolutionists have some evidence and the creationists, we think we have more evidence, and so the scale weighs in our favor. The fact is we all have exactly the same evidence. I have access to the same fossils and DNA patterns as my secular colleagues. I look at the same stars and galaxies that they do. We have the same evidence. It's just a question of how do we interpret that evidence? What does the evidence mean? What conclusions do we draw from it? And that will be influenced by our worldview, our way of thinking, our belief system. Evidence by itself doesn't really mean anything. We have to interpret evidence. And so if you come to a rock, you can't just say, well, you know, I'm just going to look at the rock and that'll objectively tell me how the rock got there and so on. No, we have a view of history, and our view of history will tell us something about the past. We can certainly use evidence in the present and try to reconstruct past events, but obviously our view of history is going to have a very profound influence on that. And you can't escape that. We all have a view of history. We all have some understanding of how the universe came to be, and that will influence necessarily our understanding of the evidence.
Take, for example, the Grand Canyon. Both creationists and evolutionists have access to the same geological features. In an attempt to explain how the canyon formed, evolutionary geologists have traditionally interpreted it as having formed over millions of years through slow and gradual processes based on the evolutionary view of history, namely the geologic timescale and uniformitarian assumptions. Geologists who accept the account of Noah's flood described in Genesis interpret the same evidence differently and describe its formation through catastrophic processes that occurred rapidly and relatively recently. This simple example describes the true nature of the origins issue. Notice both creationists and evolutionists are interpreting the same evidence and coming to opposite conclusions. This is due to the evidence being interpreted through two very different views of history. This raises an important question. If it all comes down to an interpretation of the evidence, how do we know which interpretation is correct? To answer this, we need to visit the Adirondack Mountains of New York State, where a large boulder rests alone on the 2,500-foot peak of Mount Azure. The mystery of how the boulder made its way to the top of the mountain occurred in the unobservable past. It is a historical science. No one alive today was there to verify through first-hand knowledge what actually took place. For this reason, this serves as an appropriate analogy for properly understanding the creation-evolution controversy. For the sake of simplicity, let's assume there are only two possible interpretations for how the boulder arrived at the peak of Mount Azure. The first interpretation is that Jane is abnormally strong and managed to push the 20-ton boulder to the top of the mountain all by herself. The second interpretation is that the boulder was transported by glacial ice over the top of the mountain. When the glacier melted, it left the boulder behind and is known as an erratic in geology. Now, which interpretation is correct? Without hesitation, you would immediately conclude the second interpretation is the right answer. But why was that such an easy decision to make? The answer is simple.
The glacier interpretation is most consistent with the evidence from observational science, making it the far more reasonable explanation.
The same level of clarity is possible when investigating the topic of creation and evolution.
Throughout this documentary, we will be examining key evidence from the major fields of science, including biology, paleoanthropology, and genetics. As we carefully examine the shared evidence, a key question to consider is which view of history, the biblical view or the evolutionary view, allows for an interpretation that is most consistent with observational science.
Evolution is often referred to as a very slow process occurring over many millions of years. For this reason, evolutionary scientists openly acknowledge that large-scale changes, known as macroevolution, cannot be directly observed. Naturally, this leads to the question, if it is not observable, then on what grounds can anyone claim evolution is a scientific fact? The typical response is to claim that although we have never seen macroevolution, we can see evolution happening today on a small scale, known as microevolution. Microevolution involves tiny genetic modifications that result in superficial changes in an organism, such as an increase in a bird's beak size. These type of minor changes help the organism adapt to a slightly different environment.
No one denies that this type of adaptive fine-tuning occurs through mutations and natural selection.
However, proponents of evolution believe that given enough time, the small observable changes seen in microevolution will inevitably add up to large-scale changes known as macroevolution. This is the micro equals macro fallacy, which assumes macroevolution is essentially microevolution extrapolated over long periods of time without limit. This is an example of unbounded extrapolation, which is known to be extremely poor science.
Although in theory both types of changes involve the same underlying genetic processes like recombination, mutation, selection, genetic drift, and gene flow, there's a profound and essential difference between macro and microevolution that is often overlooked. Macroevolution requires vast amounts of additional genetic information, meaning the formation of many new functional DNA sequences, while microevolution does not.
According to evolutionary theory, all living creatures evolved from a single ancestral life form that lived billions of years ago. a microorganism believed to be similar to that of modern bacteria. As the first living organism to emerge from the primordial sea, it would have had minimal genetic information, just enough to carry out the essential life functions. A reasonable estimate for the amount of DNA contained in a hypothetical primitive bacterium would be less than 500,000 genetic letters, equivalent to about 2 500-page books of typewritten information. This may sound impressive, but not compared to humans with 3.2 billion DNA letters in a single cell, the equivalent of approximately 1,500-page books. Thus, in order for an ancestral microbial species to evolve into man, a mind-boggling amount of new functional DNA sequences would have to build up over time. This new genetic information would be essential in order to specify new functions and structures such as eyes, ears, brain, heart, lungs, and a multitude of other biological features found in higher life forms. The genetic specifications for even a single type of higher life form represent a large library of information. Yet there are millions of life forms: plants, sea creatures, birds, reptiles, and mammals, each with their own library of genetic instructions.
Evolutionists claim these new genetic specifications can arise through random mutations and the reproductive filter of natural selection. Textbooks offer a number of examples that are claimed to be direct evidence of evolution in action. Let's look carefully at a couple popular examples to see whether they involve the addition of new functional DNA sequences in order for macroevolution to be possible.
Consider the observable changes occurring within canines. Wolves, coyotes, jackals, dingoes, and the numerous varieties of domesticated dog are all believed to have descended from an ancestral wolf species. In textbooks, the development of the various dog breeds is cited as a demonstrable example of evolution on a small scale. McGraw-Hill's widely used biology textbook states this type of change will inevitably lead to large-scale evolution over time. In making this claim, the textbook authors are committing the micro equals macro fallacy, the blunder of employing unbounded extrapolation. The superficial differences seen in the various dog breeds are not caused by the formation of new functional DNA sequences that are required for macroevolution. Instead, the differences are caused by degenerative mutations in a small number of already existing genes. Mutations in these genes are responsible for the variation in traits like coat color, face shape, and foreshortened limbs. But such mutations are limited in the extent of change that they can produce before they disrupt the necessary function of those genes. Factor in the well-understood process of genetic recombination, the reshuffling of pre-existing genetic information, and the formation of the many dog breeds is easily explained. Although mutations have provided an additional source of variation for breeders to select from, this is not the type of change that macroevolution requires, since it has not resulted in any new genes or new genetic information. In fact, producing dog breeds through artificial selection results in an overall loss of genetic information. The non-desirable traits that are not selected are literally bred out of the gene pool. This explains why it is possible to breed wolves and select for certain traits over a number of generations to produce chihuahuas. But you can never breed chihuahuas to get back to wolves. The DNA information necessary to get back to a wolf has been lost.
So everyone knows that within the dog kind, we have lots of different breeds of dogs. And some people might say, well, if we can see all these different breeds of dogs, you know, long ears, short ears, long fur, short fur, certainly that's an example of new genetic information that's being added to have those traits. But when we observe this at the genetic level, that is not what we see. We're just seeing variation on traits that those organisms already had. The genes that code for fur in a dog, okay, they'll produce fur, long, short, different colors, things like that, but they're never going to become wings, okay? They're never going to become feathers. They're not going to change like that because they don't have that information to be able to do that, and there's simply no genetic mechanism that allows you to add the information that you need for evolution to occur. All you see is variation within the traits that they already have.
The modern theory of evolution is founded on the concept that beneficial mutations are the raw material for eventually building new sets of genetic instructions. One of the most popularly cited examples of beneficial mutation is the development of bacterial resistance to antibiotics. Standard biology textbooks claim it is direct evidence for evolution. But is true evolution really taking place here?
Certain classes of antibiotics are considered prodrugs, which means they do not pose a threat to the bacteria until they are activated into their reactive or lethal form through the assistance of enzymes naturally produced by the bacteria. However, random mutations in the bacterium have been shown to disable the function of the required enzyme, preventing the antibiotic from converting into its lethal form, resulting in resistant bacteria. Although these mutations may be considered beneficial, since the bacteria are now able to survive the antibiotic treatments, the genetic information that encodes an otherwise useful enzyme was lost. This is the opposite direction required for macroevolution. In the long run, these types of loss-of-function mutations are counterproductive to macroevolution. For this reason, bacterial resistance to antibiotics cannot be considered direct evidence for evolution.
One of the ways in which bacteria become resistant to antibiotics is through something called horizontal gene transfer. And this is something that is pretty unique to bacteria. Humans don't do it, animals don't do it. But what happens is that bacteria can essentially swap genetic information. So genetic information can be transferred, like an antibiotic-resistant gene, from one organism to another. But simply transferring genetic information is not what evolution needs. Just transferring it from one organism to another organism, and they're both bacteria, doesn't essentially change the essence of that bacteria. So that's still not the type of genetic change that we need to go from one organism to another. We have to acquire lots of new information, lots of new functional genes to go from one kind of organism to another kind.
For evolution to create higher life forms from lower life forms, there must be a very large increase in total genetic information. Additional genetic information is needed to encode the complex biological capabilities that characterize higher life forms. Any hypothetical large-scale evolutionary change would require vast amounts of new functional DNA sequences, which would have to arise through beneficial mutations filtered by natural selection. Although adaptive changes can occur through rare beneficial mutations, it is consistently at the expense of destroying DNA information, as seen with bacterial resistance to antibiotics.
There are many mutations that are advantageous. In that sense, they're beneficial in that they help adaptation to a certain circumstance. But when they actually look at what happened, the mutation almost always involves loss of function. Something's broken, a protein isn't functioning properly. There was a very famous long-term bacterial experiment done by Lenski and his colleagues, still going on. And they documented that bacteria will adapt to a new artificial environment. You know, in a certain media, growing at a certain temperature, they can get bacteria to grow faster than original. But when they look at why those bacteria grew faster, it was consistently loss of function. You know, delete a function here, silence a gene there. And what happens is, in that artificial environment, there are a lot of normal bacterial functions that aren't necessary. For example, there's no need for the flagellum, which would help them swim, because they're in stir, so that they are agitated artificially. So they don't need the flagellum. So they can jettison unnecessary functions, and they seem to have improved, but actually they're disabled. Put them in a new environment, they won't survive. So there's a name for that, and it's well recognized within the evolutionary community. It's called reductive evolution. There's been an adaptation, but it's reduced total function.
The information-eroding nature of beneficial mutations makes sense when we think about what mutations really are. They are literally random copying errors in the DNA. Think of them as genetic typographical errors. By their very nature, they disrupt cellular processes such as gene regulation and enzyme function by scrambling the organism's genetic instruction manual. They do not create information, they erode information bit by bit. This type of genetic change occurring over deep time will never result in genome-building evolution. It is going in the wrong direction informationally. Thus, the common argument that given enough time, a series of small changes, microevolution, will eventually add up to large-scale changes, macroevolution, is a fallacy. The rarity of beneficial mutations complicates things further. It is widely acknowledged in the genetics community that harmful mutations vastly outnumber beneficial mutations. These destructive mutations are eroding the information content of the genome at a much faster rate than rare beneficial mutations could build it.
So if you have bad mutations, even if just a fraction of them are accumulating, if the deleterious mutations are accumulating even at a very slow rate, and you have very few beneficials to counteract that, you're going downhill. The fact that beneficial mutations are very rare, and that most mutations are, if they're not neutral, they're deleterious, is one of the best attested facts of biology.
In addition, most of these harmful mutations cause very subtle biological effects. They are called slightly harmful or nearly neutral mutations because their effect is too subtle to be affected by natural selection. This means natural selection can do nothing to remove them from the population. They are below what is known as the selection threshold. Consequently, these slightly harmful mutations are accumulating in the genome at a steady rate. With every generation, the average couple passes on roughly 100 additional mutations to their children. It is because of these slightly harmful mutations that mankind is undergoing genetic degeneration. In fact, there is strong evidence that we are not evolving, but rather devolving. Although natural selection can slow genetic degeneration, it cannot stop it.
The reason selection fails is mutations are coming into the population faster than they can be selected away, and because most mutations, especially beneficial mutations, are too subtle. That is, their impact is invisible to natural selection.
Genetic entropy is increasingly being recognized as a very serious problem for the modern theory of evolution. From the 1950s to the present, the apparent certainty of genetic degeneration of man has been repeatedly acknowledged by leading population geneticists, some of whom played a central role in the development of the modern theory of evolution. Contemporary population geneticist Alexei Kondrashov titled his paper published in the Journal of Theoretical Biology, "Contamination of the Genome by Very Slightly Deleterious Mutations. Why have we not died 100 times over?" To date, the problem of genetic degeneration remains unresolved, despite numerous attempts by leaders in the field to rescue the theory.
The evidence from modern genetics demonstrates that the observable changes in animals are going in the wrong direction informationally to lead to large-scale evolution. While species certainly have the ability to adapt, the extent of change is limited based on the available genetic information. Rather than one kind of creature evolving into a totally new kind through random mutations and natural selection, the Genesis account reveals that in the beginning, God created distinct kinds of plants and animals, each to reproduce according to their kind.
The first chapter in Genesis reads, "Then God said, Let the earth bring forth the living creature according to its kind, cattle and creeping thing and beast of the earth, each according to its kind. And it was so. And God made the beast of the earth according to its kind, cattle according to its kind, and everything that creeps on the earth according to its kind. And God saw that it was good." Genesis 1:20-25. If what the biblical view of biology suggests is true, that each basic kind of life arose independently, then Darwin's vision of a single tree of life with its root tracing back to a primordial bacterium would have to be replaced with a forest of trees, reflecting an independent origin for each form of life, each tree branching out into its own diverse set of species, and each tree having built-in genetic diversity, allowing for the formation of numerous variations, branches, within those basic kinds, thus enabling their descendants to survive and adapt to a wide variety of environments. This, however, is not macroevolution, since it does not involve the formation of any new functional DNA sequences. It is simply the fragmentation of each created kind into beautiful and diverse variant species through the sorting, selection, and reshuffling of already existing genetic information, just as the biblical view of biology suggests.
Since the time of Darwin, many determined individuals have searched for missing links between ape and man. Periodically, fragmentary skeletons have been found, and artists' reconstructions of their bones are presented to the public as undeniable evidence that man evolved from ape-like creatures. To scientists who study their bones, these alleged transitional forms are called homonyms.
A family tree or bush is often used to represent how hominins are thought to be related. Most of the hominin species are understood by experts in the field to be evolutionary dead ends. Apes or variants of humans that went extinct, shown as broken side branches. Paleoanthropologists generally recognize only a few hominin species in the direct human lineage. Among them are some of the most well-known discoveries, such as the famous Lucy skeleton. If human evolution is a valid theory, the hominin fossil should fall along a traceable path, starting with an early ape-like species such as Lucy into some form of Homo habilis intermediate, then into the human-looking Homo erectus and Neanderthal type, and finally into modern Homo sapiens. What you may not realize is that there are leading experts in the field that contest the status of each of these alleged hominin ancestors. Let's take a look at what we know about these hominins. Lucy, Australopithecus afarensis. Lucy is the nickname given to a partial fossil skeleton discovered in 1974 in the Afar region of Ethiopia by paleoanthropologist Donald Johansen. The bones of Lucy are believed to represent a single individual skeleton. Johansen claimed that Lucy belonged to an extinct population of ape-like creatures that once roamed Africa between 3 to 4 million years ago. The species Lucy is said to represent was named by Johansen as Australopithecus afarensis, meaning southern ape from Afar. To date, over 400 specimens have been attributed to Lucy's kind, afarensis, but nearly all of those consist of isolated bones or bone fragments that were found eroding out of the hillsides, jumbled together with the fossils of all kinds of African species. With only 20% of Lucy's skeleton preserved, it remains one of the most complete afarensis skeletons discovered. However, the bones attributed to Lucy and her kind were found disconnected and broadly scattered across the Afar landscape.
Johansen claims that Lucy is 40% complete. Well, actually, if you count the missing bones of the hands and feet, it's actually only 20% complete. So much of it is missing. Moreover, they had to sift 20 tons of sediment. So they would screen them and get rid of all the loose sediment, and they would wash it by the river, and they would pick out the bones that Johansen said may belong to Lucy's skeleton. And there were hundreds of fragments. So they had to sift not only just 20 tons of sediment, but it was an area of about 50 square meters. So do all those bones belong to Lucy's skeleton? It's an open question. In 2015, an evolutionary scientific journal showed that actually one of the vertebrae bones belongs to baboon. But for 40 years it went unnoticed, and Lucy was reconstructed in museums with that bone as a part of the skeleton all around the world. So do we really know for sure if Lucy's skeleton and the broader kind belong to the same species? Well, I don't think we should trust that, because even today, numerous experts in the field have written extensively in the scientific literature that Afarensis appears to be a commixture of ape bones and human bones. It's not a single species, it's not a valid taxon, according to these evolutionary paleoanthropologists.
It is widely acknowledged that the fossils attributed to Afarensis display a substantial range of variation in both size and anatomy, leading many paleo experts to question whether all of the remains belong to the same species. In fact, Johansen originally reported in the journal Nature that at least 2 different species from separate genera were represented in his collection of bones. He described some of his fossils as looking virtually identical to modern humans, while other fossils he described as looking distinctly like apes. Later, Johansen decided to reclassify all of his fossils, including the human-looking bones, to a single new species which he named Australopithecus afarensis. The extensive range of variations seen in his fossils were now explained as physical differences between males and females of the same species, called sexual dimorphism. To make the story work, paleoanthropologists have claimed that the larger human-looking bones belonged to males that walked upright like modern humans, whereas the smaller ape-looking bones belonged to females that lived in the trees. However, experts in the field have taken issue with Johansen's sexual dimorphism hypothesis and have cited evidence that his original interpretation was more accurate, that Afarensis is a jumble of ape and human bones.
There's 2 basic camps. There's the arborealists and the terrestrialists. The arborealists tend to emphasize the bones that have been called Lucy's kind that looked very apish. Now the terrestrialists, like Johansen, they'll emphasize the isolated bones that look more human. For example, they found a 4th metatarsal bone, and this was published in journals in 2011. And they say, see, this isolated foot bone looks human. Therefore, it proves our model, that Lucy's kind actually walked upright like modern humans, and therefore it supports the displays we see in museums and in textbooks around the world. To make sense of it, they say, well, maybe the larger bones that look more human-like belong to males. And the males walked habitually upright like modern humans, and the smaller ape-looking bones belonged to females. And so these females were more like apes and lived among the trees. We've never seen a sexual dimorphism where the males locomote fundamentally different than the females. So how do we make sense of this? Well, there's a 3rd competing view, that actually Lucy's kind is a mixture of ape bones and human bones. Therefore, it's not a valid taxon, it's not a real species. And this has actually been promoted by numerous experts in the field, including famous paleo expert Richard Leakey and his mother, Mary Leakey. In fact, even in the journal Human Evolution, Schmidt and Hausler, leading experts in the field, said just that. Afarensis, Lucy's kind, is a jumble of multiple species, not our ancestor.
In 1976, footprints fossilized in ash were found in Laetoli, Tanzania, by British paleoanthropologist Mary Leakey, and were dated to be 3.7 million years old. Researchers describe the Laetoli footprints as looking virtually identical to those made by humans. The fossilized footprints lack a fundamental characteristic of all living apes, a grasping big toe. In describing her own discovery, Mary Leakey acknowledged that they don't look any different from modern human footprints, an observation that is widely acknowledged by experts in the field. This, however, presents a fundamental challenge for evolutionary scientists, since modern humans are not supposed to have existed 3.7 million years ago. Such a finding would effectively falsify the ape-to-man theory. To resolve this conflict, Johansen and colleagues claimed that Lucy's kind formed the human-looking footprints in Laetoli, even though the footprints were found 1,000 miles away from where Lucy was discovered in Hadar and dated over 500,000 years older. The actual skeletal remains of Lucy could not substantiate their claim either, because the critical bones of the feet were missing. Soon after the discovery of Lucy, a large collection of fossils recovered at a separate site in Hadar cast more doubt on these claims. The sample included a number of hand and foot bones, including a partial foot skeleton that was assigned to Lucy's species Afarensis. In the American Journal of Physical Anthropology, evolutionary anatomists Jack Stern and Randall Sussman performed a comprehensive analysis of the hand and foot bones. It was clear to these researchers that the Hadar hands and feet were strikingly similar to living African apes. They concluded there is no evidence that any living primate has long curved, heavy-muscled hands and feet for any purpose other than to meet the demands of full or part-time tree-dwelling life. This has left evolutionists with a dilemma, which Scientific American has described as the world's oldest whodunit, an unsolved mystery.
If Lucy's kind cannot be credited with forming the human-looking Laetoli footprints, then who or what did? In an attempt to solve this mystery, former researcher of the Laetoli footprints, Russell Tuttle, suggests a far-fetched idea.
Tuttle writes in Natural History, "In any case, we should shelve the loose assumption that the Laetoli footprints were made by Lucy's kind, Australopithecus afarensis. The Laetoli footprints hint that at least one other hominid roamed Africa at about the same time." Perhaps a better explanation for why the footprints look identical to humans is simply because they were formed by humans. Tuttle admits this would be the most reasonable interpretation, except for one problem. He believes the dating methods prove the ash layers were deposited long before the first humans evolved. But what if the dates obtained were wrong? As it turns out, the Laetoli footprints were dated using the potassium-argon method, a radioisotope dating method that has an embarrassing track record of yielding exaggerated ages, millions of years, when testing its accuracy on rocks of known age that formed recently.
The major problem with all the radioactive dating methods is the first assumption, which is the initial conditions. It is assumed that when a volcano erupts and the lava flows over the ground, that all the gases escape, and when the rock cools, you only have the parent potassium in the rock, no argon. But of course we weren't there in the past to test that assumption, and that's why it's critical that we look at present-day volcanic eruptions. And one of the things that we've discovered is that the argon doesn't escape. A classic example is at Mount St Helens. It erupted in 1980, and scientists were able to watch the various lava flows building up the new dome. And a lava flow that we know erupted in 1986 was tested 10 years later in 1996, dated with potassium argon. What was discovered was that it gave an age of up to over 1 million years. Some of its minerals did. The whole rock gave an age of over 300,000 years for a rock that we observed form that was only 10 years old. And that's important because when it comes to the question of human evolution, we have to remember that the bones themselves were not dated with potassium argon. Often it wasn't the rock that they were buried in, because a lava flow would have melted and destroyed human bones. But we're looking at lava flows or volcanic ash deposits that are close to, in layers below or above where the human fossils are found. The ash that's spread out and deposited in the layer is of material that's already quickly congealed. And so that material is a prime candidate for quickly locking in the excess argon. And so the volcanic ash bed will invariably have problems when it's dated with potassium argon. And so if the potassium argon method can't be trusted on recent or ancient lava flows, then there's a real problem in establishing a true chronology for these supposed human ancestor fossils.
A more reasonable interpretation of the evidence is that the dating methods are unreliable and the Laetoli footprints look human because they were formed by humans. Handyman, Homo habilis. The next alleged hominin species on the path to modern humans has traditionally been Homo habilis. The discovery of Homo habilis was announced to the world in 1964. The bones were found at Olduvai Gorge, Tanzania, and consisted of 2 broken skull fragments, a deformed lower jawbone, a partial hand, and a tooth molar. Famous paleoanthropologists Lewis and Mary Leakey claimed the fossils belonged to the earliest toolmaker and was given the name handyman. To this day, Homo habilis is presented in textbooks as an ape-like ancestor to man. Yet few people realize there is an ongoing debate among the paleoanthropology community regarding the status of Homo habilis. Most experts in the field now deny Homo habilis a position in the direct human lineage, and many others reject the validity of this species as a classification. Much of the controversy has to do with the extremely incomplete and fragmentary nature of the remains. None of the bones identified as Homo habilis were found physically connected to a more complete skeleton. The loose bones were scattered widely across the excavated area, jumbled together in a mixed bone bed containing many different species, including monkeys, extinct apes, and humans. Some of the bones are described as remarkably like those of modern humans, and others remarkably ape-like. Paleo experts have attempted to resolve these contradictory findings by reassigning the appropriate bones to the extinct ape genus Australopithecus. To a growing number of experts, the fossils of Homo habilis look too much like a tree-dwelling ape to be considered a human ancestor. Among them is Bernard Wood, world-renowned evolutionary paleoanthropologist and foremost authority on Homo habilis, who has extensively studied the fossil remains for the past 50 years since its discovery. In Nature, Wood makes the following assessment. Even with all the fossil evidence and analytical techniques from the past 50 years, a convincing hypothesis for the origin of Homo remains elusive. The species is too unlike Homo erectus to be its immediate ancestor. Wood has come to the firm conclusion that Homo habilis is not our ancestor. Homo erectus. In the late 19th century, Dutch anatomist Eugène Dubois set off to the Indonesian island of Java. Along the banks of the Solo River, he came upon a skull cap, a femur bone, and a tooth. The leg bone resembled a modern human's, but the skull cap displayed some unusual features. Dubois interpreted the skull cap as having ape-like qualities and assigned the fossils to a new species that he named Pithecanthropus erectus. The Greek name literally translates as upright ape man, in accordance with Dubois' conviction that humans evolved from ape-like creatures. Since then, a number of specimens from Africa, Europe, and Asia have been found, and all have subsequently been renamed Homo erectus, reflecting its current status as the immediate ancestor to Homo sapiens, a claim that has not been substantiated by the fossil evidence. Most of the recovered bones are highly fragmentary, consisting of fragmented skulls, jaws, isolated teeth, and a single intact femur bone. Only one nearly complete Homo erectus skeleton has been found and nicknamed Turkana Boy, recovered in Kenya in 1984. For the first time, it provided paleoanthropologists with a clearer picture of what Homo erectus looked like. The anatomy of the skeleton and overall size of Turkana Boy is unquestionably human.
Dental evidence and unfused growth plates indicated to anatomists that had he lived until adulthood, he would have grown to over 6 feet tall. Experts in the field now universally acknowledge that the overall anatomy of Homo erectus overlaps extensively with modern humans. In fact, a number of evolutionary paleo experts insist upon reclassifying them as Homo sapiens. In their view, the skeletal features fall within the range of human variation. In addition, archaeological evidence indicates Homo erectus was able to perform sophisticated tasks and complete extraordinary feats requiring modern human intelligence, including the ability to construct a seaworthy vessel and navigate significant stretches of open seas.
Paleoanthropologists have traditionally interpreted Neanderthals as a separate subhuman species. In popular movies, they're portrayed as half-stooped, brutish cavemen with low intelligence. Few people realize this mental image stems from an outdated evolutionary perception that Neanderthals are a missing link between ape and man.
Neanderthal man. In 1908, French anatomist Marcelin Boule fraudulently arranged the bones to appear ape-like with a grasping big toe and a bent-kneed, bent-hip posture. In his view, Neanderthals were a degenerate race, too primitive to be considered Homo sapiens. Boule's errors were eventually corrected. Now, with thousands of Neanderthal specimens and a complete skeleton assembled, it is clear to paleoanthropologists that Neanderthals were human in their anatomy, albeit more robustly built, but with few relatively minor differences. Experts on human evolution acknowledge that a Neanderthal in modern dress would go unnoticed on a New York City subway. Foremost authority on Neanderthals, Eric Trinkhaus, confirms detailed comparisons of Neanderthal skeletal remains with those of modern humans have shown that there is nothing in Neanderthal anatomy that conclusively indicates locomotor, manipulative, intellectual or linguistic abilities inferior to those of modern humans. Archaeology has further revealed an impressive cultural inventory, including fire, stone tools, tailored clothing, art, musical instruments, cosmetics, jewelry, and ceremonial burial of their loved ones, a defining aspect of what it means to be human. In 2010, Swedish geneticist Svante Pääbo and his international team of researchers sequenced the first ever nearly complete Neanderthal genome, confirming once and for all that they were fully human and members of our own species, Homo sapiens.
About 3% of my genome is Neanderthal. About 3% of your average Eurasian is Neanderthal. But if 60% of Neanderthal is amongst Eurasians, that means that we all have different pieces. When they discovered DNA in Neanderthal bones, that wasn't supposed to happen. The bones are too old. In the evolutionary model, 30,000 years ago, 50, 100,000 years ago, Neanderthal bones should have no DNA left in them. And yet they do. That was a big surprise. And when we look at the ancient DNA we can pull up and sequence DNA, it's clear multiple interbreedings happen between human Homo sapiens and human Neanderthal. They're not supposed to be human. And yet, 3%. That means that for every 100 of my ancestors, 3 of them were a Neanderthal. That makes them people. Homo sapiens. Human beings. And so therefore should not surprise us about the fossil record. All these things we're learning about Neanderthals recently has completely changed the picture that we were given when we were younger, especially the picture they tried to give us in the 1800s.
The age-old evolutionary assumption that Neanderthals were a subhuman species has been disproven. Evolutionary paleoanthropologists traditionally expected the hominin fossil record to reveal a traceable linear progression where an ape-like Australopithecus species, such as Lucy, evolved into Homo habilis, which evolved into Homo erectus, which evolved into Homo sapiens. In recent decades, however, this view has been dramatically overturned.
Instead of a simple human family tree with a few branches, the paleo community now describes the hominin fossil record as a messy bush, with many disconnected branches showing no clear evolutionary progression from ape to man. The pattern that is emerging in the hominin fossil record is surprisingly consistent with a biblical view of human origins recorded in Genesis, which teaches that humans and apes were created as independent forms of life. Indeed, there appears to be a clear separation between two distinct types in the fossil record, the human type, called Homo, and the extinct ape type, called Australopithecus. These two types are found together in rock strata of equivalent age, as far back as hominin fossils are found, just as the Laetoli footprints have confirmed. This shows that humans have always coexisted with a diversity of apes, rather than one evolving into the other.
It's hard to find anyone who has not heard the often repeated claim that humans and chimpanzees are genetically 98 to 99% identical. This has been promoted to the world as proof that humans share a common evolutionary ancestor with chimps. However, recent studies now challenge this claim.
Evolutionary geneticists have acknowledged that the actual genetic differences are far greater than we've been told. For example, primate evolutionist Todd Prius states, "It is now clear that the genetic differences between humans and chimpanzees are far more extensive than previously thought. Their genomes are not 98% or 99% identical." Earlier studies published in the evolutionary scientific literature reported an overall DNA similarity of 98 to 99%. However, large portions of the chimp genome did not align with the human genome, and so were excluded from the reported estimates. For instance, the algorithm parameters used in the major milestone publication in Nature reported by the Chimpanzee Sequencing and Analysis Consortium omitted over 100 million DNA letters. When accounting for these large non-alignable regions and other omitted sequence data, the actual chimp-human DNA similarity is significantly lower than the 98 to 99% identity claims.
When they originally started to ask that question, they didn't have full genomic data, and so they're actually based their estimates of similarity based upon just little snippets of the genome. And they were choosing protein coding sequences which are the most similar, that explain why we have similar biochemistry. So initially we're saying, 98, 99% of the genomes must be similar between chimp and human. The textbooks still say that, but basically geneticists know that's not correct.
What is the actual genetic difference between humans and chimpanzees? This is a question that comes up over and over again in this creation-evolution debate. And if it is true we're 98 to 99% genetically identical to the closest evolutionary relatives, evolutionists say, it feels like they're just kissing cousins. Well, let's dig down to what the scientific data actually say. If you look at the 2005 chimpanzee genome paper in Nature, you look at subsequent papers, the bonobo paper, the gorilla paper, the orangutan paper, all of them give basically a consistent answer. That most of our DNA can be aligned to chimpanzee and vice versa, and in that region that does align, it's about 1 to 2% different in terms of single letter differences. But if you say, wait a minute, are there sections of human DNA that fail to align to chimpanzee? Are there sections of chimpanzee that fail to align to human? The answer is yes, and it's far more DNA than is that 1 to 2% difference. So if you incorporate all of these numbers together, the stuff that aligns and is almost identical, the stuff that can't be aligned at all, you get a number about 85%. What's even more important is what that percentage translates to in terms of absolute differences. So in 85% identity, 15% difference in terms of raw DNA letters represents 300 to 400 million single DNA letter differences. That's a massive number.
The evolutionary theory claims that humans evolved from a hypothetical chimp-like ancestor roughly 6 million years ago. This is said to have occurred through a long series of beneficial mutations. In light of the actual genomic differences between humans and chimps, this is simply not genetically feasible.
A more accurate chimp-human DNA similarity estimate of 85% represents 300 to 400 million DNA letter differences, an extreme level of genetic discontinuity. This means in order to evolve a chimp-like ancestor into modern humans, hundreds of millions of beneficial mutations need to arise in an ancestral population. The difficulty with accomplishing this has to do with the extremely long waiting time required for establishing even the smallest sets of mutually dependent mutations. Even granting a best-case scenario for evolution by generously assuming human and chimp DNA is 99% identical, the remaining 1% would still be a difference of 30 million DNA letters, an impossible genetic barrier for evolution to traverse in 6 million years.
Let's assume that it's still only 1%. That's not correct. No one believes that now. But even if to get 1% of the genome different requires 30 million mutations, that's a lot of new information. And when we talked about the waiting time, we were saying, well, 8 mutations, waiting for 8 specific mutations takes more time than the time since the Big Bang. So, no, it's not conceivable. You cannot change the program of an ape into the programming for a human in any amount of time. People ask, well, what's the problem? You're saying there's 100 new mutations per person per generation in a big population. That's billions of mutations every generation. What's the problem with getting the information that codes for something? Well, the difference is that genetic damage is non-specific. So deleterious mutations can happen in anywhere in the genome. There's no specificity. So creating damage is easy, but in the case of a manuscript or a genetic program or a computer program, it's easy to break them by changing letters. It's really hard to improve them. And you have to wait a really long time for the specific letter to mutate into the specific alternative letter at a specific site to actually create any type of benefit. So waiting time for beneficials is different from waiting time for neutrals. You don't have to wait long for a neutral or for a deleterious. You have to wait a really, really long time if you're waiting for a specific and beneficial one.
Waiting for the right mutations to arise and become established in a pre-human population greatly exceeds evolutionary timescales. Leading evolutionary geneticists acknowledge it is a serious problem for the theory, devastating for the ape-to-man scenario. Population geneticist Michael Lynch confesses in the Journal of Molecular Biology and Evolution. A central problem in the evolutionary theory concerns the mechanisms by which adaptations requiring multiple mutations emerge in natural populations. Lynch's calculations suggest the length of time required for just 2 specific mutations to become established in a pre-human population is over 200 million years, well beyond the 6 million year time span during which an ape-like creature is said to have evolved into man. Other studies reporting in scientific literature show similar results. Evolutionary geneticists Durrett and Schmidt of Cornell University report in the Annals of Applied Probability that the average waiting time to form a slightly longer DNA sequence of 8 specific mutations is on the order of 650 million years. But this estimate is incomplete. When accounting for random loss due to a well-established principle known as genetic drift, the actual waiting time should be 100-fold longer, roughly 65 billion years. This is 4 times longer than the reputed age of the universe, assuming a Big Bang singularity 13.7 billion years ago. At best, all evolution can hope to accomplish in the prescribed 6 million year time span is the formation of a tiny DNA sequence, no more than a few genetic letters in length, totally incapable of producing a single new gene. Modern genetics has demonstrated that it is impossible for humans to have evolved from a chimp-like ancestor via random mutations. It is an unbridgeable genetic gap for evolution to traverse, even given billions of years.
If humans did not evolve from ape-like creatures, then where did we come from? The Genesis account of creation states that God made Adam and Eve. not as mythical beings, but as the literal historical ancestors of all living people. They were the first two human beings created, with all humans descending from this first couple. From a purely genetic standpoint, is it scientifically feasible that all humans descended from a single mother and a single father? Many insist this is absurd, and yet the genetic evidence for a literal Adam and Eve is inside each one of us. It's found in our DNA.
In 1987, a milestone paper was published in the journal Nature by leading evolutionary geneticists who announced the results of a mitochondrial DNA analysis. Geneticists from the University of California found that all humans are descended from one woman thought to have lived in Africa 100,000 to 200,000 years ago. Their results sent shockwaves throughout the scientific community and called for a major rewrite of the evolutionary view of human origins to accommodate the new data. The revision gave rise to the now widely accepted out-of-Africa theory. Proponents of the theory couldn't help but notice its uncanny resemblance to the biblical Eve. In acknowledgement of this, they gave the genetic mother of us all the name mitochondrial Eve. According to the evolutionary perspective, mitochondrial Eve was an unnamed woman who evolved out of Africa from a Homo erectus population of ape men. Not long after the first mitochondrial DNA studies revealed a single mother of us all, evolutionary geneticists found similar results when analyzing sequences on the male Y chromosome. In 1997, a team of researchers from Stanford University reported to the American Society of Human Genetics that all men inherited their Y chromosome from a single male ancestor. The sequencing of thousands of Y chromosomes from diverse people groups living around the world has revealed an overall lack of Y chromosome diversity. All men share the same Y chromosome, plus a small number of mutations consistent with a single male ancestor of the human race. Once again, evolutionary geneticists couldn't help but give the father of us all a biblical name, Y chromosome Adam. Just as they did with mitochondrial Eve, evolutionists interpret Y chromosome Adam to be an evolved ape man from Africa that lived sometime around 100,000 to 200,000 years ago.
The evolutionary community acknowledges that there is a literal Y chromosome Adam and there is a literal mitochondrial Eve. They say it's clear that all of the Y chromosomes on this planet trace back to a single individual who didn't live so long ago. The Y chromosomes only passed on through the male, right? Father to son. Well, the mitochondrial chromosome is only transmitted through the female. And that chromosome also, all the geneticists agree, all people on the planet get their mitochondria from a woman who lived not so long ago, and it's uncontested. And it turns out initially the evolutionists said, yeah, but it looks from our estimates of how long ago those individuals lived that they didn't live in the same time span. Now actually that they've reworked their numbers, they're always reassessing their time scales. They're Adam and Eve. The mitochondrial Eve and the Y chromosome Adam, they are in the same time span.
There are two fundamental differences between the Adam and Eve of the Bible and the evolutionary interpretation of mitochondrial Eve and Y chromosome Adam. The first difference has to do with time, and the second has to do with population size. The Genesis account indicates Adam and Eve lived recently, just thousands of years ago, and that they were the only two people alive at the time of their creation. The evolutionary model claims they lived around 100,000 to 200,000 years ago and belonged to a hominin population of 10,000 individuals. But as you will see, it's not the genetic data that conflicts with the biblical account of Adam and Eve. The conflict comes from inferences about time and population size.
Evolutionary geneticists estimate that genetic Adam and Eve lived around 100,000 to 200,000 years ago using a method known as the molecular clock. The technique relies on the assumption that mutations accumulate in certain regions of the genome at a constant rate over deep time.
Evolutionary scientists have to further assume that humans evolved from a chimp-like ancestor roughly 6 million years ago in order to calibrate the molecular clock. Both claims are problematic and have been called into question by the genetics community. Distinguished evolutionary geneticist David Reich of Harvard confesses in the publication Nature, "The fact that the clock is so uncertain is very problematic for us. It means that the dates we get out of genetics are really quite embarrassingly bad and uncertain." Scientists are now using a more straightforward approach to determine mutation rates that do not require ape-to-man evolutionary assumptions. It involves directly measuring mutation rates in the present, comparing parents and offspring, known as the pedigree method. When comparing DNA sequences between parents and children, the measured mutation rates are typically 10 to 20 times higher than those inferred based on assumptions of ape-to-man evolution. When the molecular clock is calibrated using the empirically measured mutation rates, both mitochondrial Eve and Y chromosome Adam lived just thousands of years ago. In discussing the age of mitochondrial Eve, evolutionary scientists in Trends in Genetics acknowledge this discrepancy. mtDNA datasets often exhibit anomalous patterns. One of these anomalies is the discrepancy between mtDNA mutation rates observed in evolutionary timescales, for example, in dating the divergence between two species and those measured within family pedigrees. If the high mutation rates seen in some human pedigrees were used to calculate the age of our most recent female common ancestor, she would have lived just 6,000 years ago, a date more consistent with biblical Eve than mitochondrial Eve.
There's a long-standing debate and argument in genetics, and that deals with the difference between what's called the phylogenetic mutation rate and the genealogical mutation rate. If you look at a family, you can count the differences between the people and you say, oh, they've had some mutations. And you can actually calculate a mutation rate over time in today's time, in known time. Right now, we know what the mutation rate is. But the evolutionary community doesn't like to use that rate because it's too fast. They use what's called the phylogenetic mutation rate. That is, they look at the difference between humans and chimpanzees, not the real rate, but the differences, and they say, oh, we've been separated for so many millions of years. And when I was an undergraduate, that difference was 3 million years. Now it's 6 million years, 6.5 million years. Some people are arguing for 13 million years. Because mathematically they're having a hard time dealing with this. Take the differences between humans and chimpanzees in some gene, like mitochondria, Y chromosome, or something like that. Divide it by the time that separates us, 6 million years. You get a very slow mutation rate. But if you look at the people alive today, count up the number of differences between them, and divide by the time since their ancestor, maybe their great-great-grandfather or something like that, and you're going to get a mutation rate much faster. When we use that rate, Adam and Eve fall into the biblical time frame. So when we use today's science, measurable science, laboratory stuff we know is true, Adam and Eve are biblical.
While the actual genetic data reveals a single mitochondrial Eve ancestor and a single Y chromosome ancestor, evolutionists have insisted they must have lived in a larger population. For this reason, they caution the general public not to mistake the evolutionary Adam and Eve with Adam and Eve of the Bible. Yet they acknowledge there can be no direct evidence for the larger population size of 10,000 because all other mitochondrial and Y chromosome lineages supposedly died off, removing all traces of their existence, leaving, as paleo expert Chris Stringer has said, just one lucky mother and one lucky father. In an attempt to explain what would otherwise be another remarkable biblical coincidence, proponents of the ape-to-man model invoke what is known as coalescent theory. This theory assumes that over many generations, it is inevitable that eventually only a single mitochondrial and Y chromosome lineage will remain.
Coalescence theory is based upon the assumption of random mating. So you need a single population and all the people have equal likelihood of intermarriage. That's never been the case. From an evolutionary point of view, humans and prehumans have always been in tribes that are separated. So there's not random mating. In fact, if you look at the charts that show coalescence, all you have to do is say, okay, that's one subpopulation, but there's dozens or hundreds of other subpopulations, and those would be producing a different coalescence to a different Adam or a different Eve. Basically, the coalescence argument fails. So their rebuttal position is not scientifically valid, and it's relatively reckless.
Evolutionary scientists reasoned that if humanity started with just 2 people, it could not explain the total amount of human diversity we see today. For this reason, a population of 10,000 became a central tenet of the current model.
It's typically assumed that Adam and Eve would have had no genetic variance, no genetic diversity. However, there's no reason to assume they were created as nearly identical clones. If Adam and Eve were created with built-in diversity for traits such as skin color, all of the different-looking people groups could easily arise in a biblical time frame. There would be no reason to wait millions of years for the slow accumulation of mutations if the diversity was encoded in their genomes right from the beginning.
If Adam and Eve were heterozygous, as we would reasonably expect, extensive genetic diversity would quickly arise simply through sexual reproduction, which reshuffles pre-existing variation to produce different combinations of traits.
This idea that God creates an initial pair with differences between them and within them. We'll call that term, the technical term is heterozygosity. So this is an idea that has massive consequences in modern genetics and virtually all of our genetic discussions. It also provides a convenient explanation, a very plausible explanation for how you get all the ethno-linguistic differences that we see today. Humans are considered one species, and we can produce variety in a single generation. So if a dark-skinned Sudanese person marries a light-skinned Finnish individual, their children will be an intermediate skin tone and have features from both parents. And if you had a Australian Aborigine who marries a Han Chinese, and they had children, and so then the offspring of these 2 mixed marriages had children, they could produce a whole diversity of individuals. Why is that? Because people today still have heterozygosity. They have less heterozygosity than what I'm saying Adam and Eve had. In other words, Adam and Eve could have produced in their many children every version of human that we see today. Once you have an original pair, original population, an original set of 8 if we're talking about the flood, that has heterozygosity, you can explain people groups just like that.
A striking example of built-in genetic variation can be seen in the example of twins born from mid-brown parents. Mother Kylie Hodgson and father Remy Horder gave birth to twins Remy and Kian, who appeared racially different. The 2-tone fraternal twins shared the same womb and were born a minute apart. What most people would typically see as racial differences arose in a single generation due to gene segregation. Just like people living today, one can reasonably expect Adam and Eve to have had a large pool of genetic variation built in to their genomes. Prior to modern genetics, evolutionary scientists claimed that there were fundamentally different races of humans that evolved in diverse parts of the world over a long period of time. This model was known as the multi-regional hypothesis. This perception changed with the advent of DNA sequencing, which revealed that all human beings are remarkably similar genetically. It is now well established among the scientific community that all people, regardless of their skin color or ethnicity, are 99.9% genetically identical. The so-called racial features that we tend to focus on are essentially only skin deep, reflecting trivial differences in our genetic material. Physical traits like eye shape and melanin production amount to a minuscule 0.012% difference in our DNA. This means that regardless of our cultural differences, genetically, we are all part of one big human family.
If you have designed organisms, created organisms, they would be designed and created so they have internal diversity. You don't have to wait for mutations. So the waiting time goes away. If you want adaptation without deep waiting time, then just design the organism with variants in it. Even a single organism has heterozygosity, and so there's lots of genetic diversity, even in a single organism. Two individuals have 4 chromosome sets, and they have the ability to basically accommodate every possible variant. So 2 people is enough to allow for vast amount of genetic diversity that could be used by natural selection, could be used for adaptation to new environments, and you don't need deep time to do that.
The findings from modern genetics have compelled evolutionary scientists to develop a revised story of human origins that shares many striking similarities with the biblical account of the Tower of Babel dispersion. The new model is called the Out of Africa theory and has been popular for many years. This theory proposes there was a near-extinction event causing a population bottleneck that reduced humanity to a single breeding population of just a few thousand survivors.
In the Out of Africa theory, which is now on the rocks, but in the Out of Africa theory, they postulated there was a human population of about 10,000 people for an indeterminate amount of time. They never really say how long it is, but let's say 10,000, 100,000 years. If we compare a modern species that has about that many individuals, you can look at the African cheetah. There's about 10,000 of them in the wild, give or take a few thousand, and they're having massive problems. Birth defects are increasing, reproductive incompatibility in couples is increasing, litter size is decreasing. All the population biologists are essentially assuming cheetahs are going to go extinct. How did Homo sapiens evolve? We supposedly went from Homo erectus to Homo sapiens. You know, Homo erectus is smashing rocks together, and Homo sapiens that can fly to the moon during a population bottleneck that should have driven us to extinction.
This theoretical near-extinction event has been used to explain why all humans are nearly identical genetically. The Out of Africa theory claims all living humans originated from this small population who lived in the general vicinity of northeast Africa. Alternative studies suggest the small founding population was a little further north, in the general vicinity of the Middle East. The survivors of this theoretical near-extinction event suddenly grew very rapidly, splitting into numerous smaller tribes which dispersed outward and gradually filled Europe, Africa, Asia, and eventually the Americas. Each initial tribe gave rise to its own language and culture, and continued to split and disperse to form the people groups and languages we see today. Each migrating tribe would have carried with it a different sampling of the original population's gene pool, rapidly producing the distinctive features that some used to define race. In this model, mutations would occur but are not necessary to explain these distinctive features, which means deep time would not be required either.
You know, just using standard population genetics principles, we can explain the origin of races easily in hundreds or thousands of years. All we need is the population to fragment into smaller populations. Isolation by distance is going to cause changes over time independently in each population, either through natural selection or just random drift. So we have a model for that at the Tower of Babel. A few hundred years after the flood, God separated the nations according to Y chromosome. It's the male lineages that define each population, and they spread out on the earth, and they would have remained separate from one another because of language differences for a time. That's all we need. Once you do that, you're going to have regional differences amongst the people. So all of a sudden we have people start looking more Asian, people that start looking more Norwegian, people that start looking more Australian Aboriginal, and people that start looking more African. All we need is isolation in a short amount of time.
Evolutionary geneticists reporting in the proceedings of the National Academy of Sciences have named this specific model the instantaneous divergence model. This model is virtually indistinguishable from the biblical model of the human dispersion associated with the Tower of Babel as recorded in Genesis chapter 11. Now the whole earth had one language and one speech.
So the Lord scattered them abroad from there over the face of all the earth, and they ceased building the city. Therefore its name was called Babel, because there the Lord confused the language of the whole earth. And from there the Lord scattered them abroad over the face of the whole earth.
One of the funny things about the out of Africa theory is that it actually mirrors the biblical account. We have a population of people that originated in Africa sometimes in a distant past. Well, biblically we started with 2 people. Our population grew to thousands of people, we don't know how many. And then our population crashed biblically at the flood. Well, in the out of Africa theory we had a lot of people and it crashed during a bottleneck. And then the population began to grow in both models, and then the population fragmented and spread across the world in both models. The Bible was written first, and we're stuck with it. There's a limited range of possibilities biblically about human history. Back in the day, though, evolutionary theory, they could say whatever they wanted, and the most common view back in the day was the multi-regional hypothesis, that human beings just evolved in synchrony across the planet and exchanged genes as we evolved and evolved and evolved. They did not expect a bottleneck. They did not expect going through the Middle East. They did not expect a Y chromosome atom and a mitochondrial leave. But those are things that have to be true if the Bible's true. And those are things that the modern geneticists have discovered. So what we're seeing over time is that the evolutionary model is getting more biblical, which is really interesting.
Throughout this documentary, we have examined key evidence from the major fields of science, including biology, paleoanthropology, and modern genetics. We have seen that the evidence from real observational science is consistent with the major origins-related events described in Genesis. The tide is changing. Scientists and scholars are beginning to see Genesis as far more than a book of myth, but as a reliable account of our origins. Thanks for joining us on this fascinating journey through science and history. I look forward to seeing what new discoveries science will bring and the impact it will have on the creation and evolution debate. Until next time, I'm Rebecca Tadrove.